Thank you. I'd like to thank the people who nominated me for this award, and it's very much appreciated. The blood methodology is very statistical, and I could give you lots and lots of formulas, but after lunch, everyone will fall asleep. So I will not give a lot of formulas. So I'll just give you sort of my history and where it has led me through my career. And it covers Sweden and many other places. So my history goes back to what was once called Prussia. My ancestors came from that area of Europe, and they came to the US in the 1860s. And these are my grandparents, Edwin and Clara Lawrence. And since I was born, I was under the assumption that this was how my family tree looked. My grandparents had 8 children. There's only 7 there. 1, of course, did not survive. Fran is my mother, and Edward was my father. And I have a sister who was adopted some years later. I was the first grandchild of my grandparents as well, so I was very spoiled. My grandparents had a dairy farm. About 30 cows is all they had, but it gave me my urban background for my life. They had 1 red and white Holstein, and the rest were Holsteins. They received the Hordes Dairyman magazine. It came every week, and I read through it a lot and read about bull proofs and cow judging. And there were lots of ads in these magazines, so I sent away for everything that was free. Most of it wasn't very useful, but so it was in their dairy farm where I learned more about dairy cattle and looking at differences in yield. It didn't really matter to my grandfather as long as they gave milk, and he also was involved in raising crops. He actually wanted me to become a corn breeder, but I defied him and became an animal breeder. During my high school years, I had a biology course, and I learned how to cross flowers. So I attacked my mother's garden, and the next year I got different kinds of patterns in the petunias, and my mother was upset with me for that, for ruining her patch. So, and she didn't even like it when I did this. So after that, I convinced her I should have a mouse. So we went to a store and bought a mouse. They pulled it out of this fish tank with lots of mice. Here's a male. It will not cause you any problems. A week later, after we got it home, we had a litter of 6. And I was into raising mice. So we went out. We actually drove about 40 miles to a breeder who had other than white mice. So we bought a black male, and I crossed it to one of my females and ended up with a lot of different colored offspring. Some were silver with red eyes, some were red colored with red eyes, some were red with black eyes. I kept pedigrees for all these mice, but at that time I didn't know anything about inbreeding, and I knew some of them were inbred. I was getting crooked tails and things like that. So then I went to undergrad at Purdue University, 65 to 69, and in my first year I took a genetics course where they taught you how to do experiments with fruit flies for crossing over and determining map distance. Also with flower beetles and Naraspa crassa. And the professor there was Dr. Wayne Keim. And after this course, I began working for him. And so my job was to propagate the fruit fly lab. And he also had grad students who worked in bird's foot trefoil, alfalfa, and chickpeas. I had to grind chickpeas for ages. But the fruit fly lab was neat because he had about 50 different varieties with different mutations. And during my propagating, I did my own experiments. I was able to get some vampire flies, so I called them. They had black bodies and white eyes, and they had wings that looked like teeth. So that was my fun during undergrad. In '69, I went to Cornell, and these were my office mates. Jeff Kewen wasn't my office mate. He had an office on the next floor up, but he was mostly in our office. He liked to socialize. Jorge Velasco was from Peru, and Ivan Mao got his PhD and went to Michigan State University. And he did a sabbatical here in Denmark. And this was our group in those days. And there's Dr. Henderson, CRH. We had a visiting statistician from Iowa State, and Bob Everett, who patented the test day model, and Dale Van Vleck, who's at the University of Nebraska. Of course, he's retired. But that was the entire reading group. There's also Brian Kennedy, whom some of you might know or have heard of. He was from Quebec. He went back to Quebec after he finished his PhD, and then went to California, Davis, and then eventually came back to Guelph. And we did a lot of collaboration on work. And in 1994, after the World Congress in Guelph, he passed away from cancer. So that was a loss for us. So at Cornell, Dr. Henderson developed what was called the Northeast AI sire comparison using a BLEP sire model. And he wrote a bunch of programs which created the non-zero coefficients of mixed model equations. And he was able to solve for the sire estimated transmitting abilities. So he ran these programs himself the first 3 times. And Ivan Mao and myself, we learned how to run these programs as well. And during Christmas, between 71 and 72, Ivan and I were left with the responsibility of running the programs. And you could only run programs at Cornell from 11 p.m. after the DHI people finished with the computers, and then they would take over again at 8 in the morning. So we only had the evening hours to do this. And at that time, the sires were assumed to be unrelated. So A is the relationship matrix, and it was fun for Ivan and I because we ordered in pizza, and everything was on magnetic tape then, and so there were 8 magnetic tapes, and as they ran, the lights would flash, so we'd turn out the lights and watch the lights on the tape drives run. So in 73, I finished. I actually went to Fort Benning, Georgia, did 3 months in the military. And that was when the Vietnam War was coming to an end, so they didn't want people anymore. So they let me come to Guelph as a postdoc. Less than a year later, I was able to apply for a faculty position at Guelph. I had to leave Guelph while that was in progress because I had to become a permanent resident. So I went back to Cornell just for fun. And in that year, Dr. Henderson developed his A-matrix inverse procedure, which was a very fast way of inverting. And so we implemented the relationship matrix into the sire model at that time. And then 1976, Knut Ronigan was head of the group here, and he invited Dale Van Vleck to come and tell them about the BLUP-sire model and the programs. And Dale either was busy or didn't want to do it, I don't know which, but he told Knut to invite me. So I ended up coming. And I got in the newspapers here. Could never read the article, but I demonstrated the use of those programs and taught people what BLEP was, made some notes, which ended up being distributed everywhere. Uh-oh. And at the end of, I was here a couple of months. At the end of it, I gave a 1-week course. And this was the group that attended. This is Knut here. And Britta Donnell is next to me. And I think Jan Philipsen's behind her. Jan Dommerholt of the Netherlands. Pim Braskamp of the Netherlands. Erling Finland from Norway. And Eildert Groneveld from Germany. And there were some people from Finland and Iceland, and I didn't learn everyone's name in the short week. So it was about 1979 that I met my wife, Laura, and she went with me to see my parents once. And at that time, I learned that I was adopted too. So I was not related to any of these people at all, which is a weird feeling after 33 years. But it was something new that I had to contend with. Because in the later years now, I've had troubles with diabetes and heart fibrillations, and the doctor keeps asking me, well, what did your grandfather have or your father? And, well, I'm not related to any of these people, so I don't know what my background is. So it was sort of a dead end for the doctor. 1985, I gave a course in Norway, and there's Britt Bergeland, Eva Marie Stahlhammer, Erling Strandberg, Ulf Emanuelsson, and maybe others from Sweden. I ended up, over my career, giving 17 of these kinds of courses, going from a week to 2 weeks. And the largest group that I had was in Munich. In 2010, there were over 60 students, plant breeders and animal breeders. So after that, I developed a technique for solving mixed model equations that was easier than what we were doing, called iteration on data. And in 1989, people started using animal models because computing hardware had come along. and made things possible. And then estimating variances, the heritabilities and genetic correlations, we use Remmel and Bayesian methods. And then we also had to deal with animals whose parents were unknown, which happens when animals change herds often and when they're imported from another country. So we had to implement a phantom parent grouping strategy. And then after we get bull proofs, people wanted to start comparing bulls from different countries. So they used a conversion formula. Country A, their estimated breeding value was equal to a regression, a mean, small a, plus a regression on the EBV in another country. And Hans Wilmich and Mike Goddard both published methods for doing this. It wasn't so much the methods that were the problem, it was the data that went into them. So when you're using bulls from country B in country A, because of the high semen price, you don't randomly mate them to cows, and their semen is very expensive. So they don't get used the same as a bull within that country. And a lot of countries did some weird science on how to do these things. So when you're comparing two countries, at that time there were hardly 20 or more bulls that could be used to compare different countries. And this procedure only allows you to do two countries at a time. So in Guelph, we have Cmax Canada at that time. We have the milk recording people, and not too far away we have the Holstein Association. So Cmax became upset with conversion formulas, and so they come to my office, Larry, do something, help us. And I tried to, for the longest time, to stay out of international comparisons because to me, it wasn't really sound science. And it was very politically charged, and I didn't know if it would be very rewarding in the long run. But anyway, after they asked for help, I had a meeting with Jacques Chenet, who ran the genetic evaluations in Canada at that time, Ted Burnside, who was a colleague, Herman de Boer, who worked for Cmax Canada, and Wenchan Zhang, who was a postdoc with Ted. So we had a meeting at Cmax, and I outlined a model which allowed for many countries to be analyzed at the same time, and allowed for the fact that bulls were not perfectly correlated in their daughter performance between different countries. And so after they agreed to it, we got some data from the Netherlands, US, Italy, and Canada, and I did a test run at that time. In 1994, presented the results to an interbull workshop in Denmark. And after that, Yorgos Binos, who was a director of operations here at the Interbull Center, he seemed to adopt it immediately. I gave him all of my programs. And as far as I know, it is still going. And only now it's called Gmace because we're incorporating genomics. And they're analyzing 1 billion traits. And the whole thing has sort of taken on a life of its own after I gave Yorgos the programs. And I've lost track of all the changes that have been made over the years. So I'm not sure if it helps Cmax or anyone. There were there's always changes going on, but it was at least a little bit better than the conversion formulas. So in 2000, 2001, people started genotyping dairy bulls, and they were using that because the dairy bulls had lots of progeny and were well evaluated. And Mewison and Goddard wrote a paper on how those genotypes could be used to select bulls earlier. So as soon as they're born, you genotype them, you can get a breeding value that's more reliable than a parent average of the parent breeding values. So since then, there's 50K chips and 200K chips. They just keep getting more and more SNPs that you can genotype animals with. And then imputing, if you only did a 50K evaluation, you could impute up to the 200 or 700. So there was lots of that going on. US and Canada agreed to have a project where we genotyped a lot of dairy bulls. And now they have a 10,000 cow project. But in Canada, we're getting many, many cows being genotyped now. So we have lots of genotypes on female animals. So based on the Mewison and Goddard paper, I presented a method to the Canadian AI industry. We usually had 2 meetings a year with the industry, and I showed them how they could double their rate of genetic change, and they could save 95% of their costs of progeny testing. And I did that in a 30-minute talk. It meant they'd have to cut back the number of bulls quite a lot, and they just huffed and laughed and didn't believe it could be done. And they said CDN would lose a lot of money if the number of bulls tested went down. AI would have a lot of unused barn space if we didn't test as many bulls. They'd have to lay off workers, so they didn't want to have anything to do with this. So I was a little frustrated, so I published it in 2006, mainly to prevent people from patenting the method. The rest of the world seemed to understand the possibilities, and of course today Cmax is doing exactly what I said they should do back then. And they're making lots of money. But also now, because of genomics, they're becoming more possessive of their genotypes on their animals, so they're not willing to share them as readily. And they're also contemplating not using Interbull and CDN, which is our genetic evaluation center in Canada. So they're actually computing genomic EBVs within their own company and using those for selection more than outside information. And of course, the cost of genotyping keep coming down, and now everyone's getting into sequencing the genome. So since they were genotyping bulls, I decided to genotype myself. So I sent a DNA sample to a company called 23andMe, $90, 2010. I thought I'd find out something about my background, and they also screened you for hundreds of different diseases. So maybe that'll help my doctor anyway. So I gave all the results to him. He ended up retiring, so now my new doctor doesn't know anything. But luckily, nothing else really showed up on that. I'm 2.4% Neanderthal, which explains a lot of things, I think. And 0.2% African, but most of the rest of it is from Northern Europe, which is the Prussia area. So now, after that, when I sent to Illinois Public Health to find out who my birth parents were, because they had the original birth certificate. And it turns out that my biological mother was what I called my aunt at that time. She became pregnant at the age of 15, and at that time, in those years, getting pregnant while not being married was not a good thing. So they sent her to Chicago to a home for unwed mothers, and that's why I was born in Chicago. And I was adopted by her sister, Fran, which I called my mother. So my mother is really my aunt, and my aunt is really my mother. And after I found out this, it explained a lot of things that happened early on in my life. But anyway, so now 63 years later, I know where I am related to all these people. My grandfather had diabetes. My grandmother lived to be 94, so hopefully that's good for me. So that's somewhat a story of genetics, messing with my mother's flowers, going to my grandfather's farm, raising mice, crossing fruit flies, giving a course here in Sweden, Uppsala. Oops. I have to say that I've been very lucky and fortunate to meet so many people in animal breeding over the years. That course here in Sweden opened me up to a lot of people in Europe, and I went to EAAP meetings. I was also fortunate to have gone to Cornell and learned about BLUP and the sire model and the programming. And I was fortunate to have visited Sweden when I did, fortunate to have worked in Guelph, next door to all these associations. So you have to make the best of what you have and what you know at the time. And in the future, you have to think where things might go. For example, maybe in the future, every new animal that's born will be genotyped or sequenced. So we should be planning for that. We might want to use something other than an animal model in the future. And so right now people are using single-step GBLUP. So I think that'll be replaced fairly soon. So there's more need to estimate SNP marker effects more precisely rather than the genetics of individuals. And then we can look at whether SNP marker effects differ between populations or countries. If you're a student, you should go to meetings and attend presentations by other well-known scientists, even if they're in a different field. They always have ideas that might affect you. Some people like to keep a list of ideas in their notebook. I do that, but then I lose the notebook. And have an open mind, go to other countries, such as Sweden and other institutions. You learn different ways of doing things. And at this stage, since I'm retired, I try to keep my mind active and help others as much as I can. So, thank you.